A lot for the introduction. So, uh, to come to this topic that we have going on for past 6 days, it's the tailored medicine or personalized medicine. So, this idea that I got from was when I was doing my PhD and my research, where we go through lots of complications, like say we have lots of disease that comes from because of medicine. So when I was there in Vienna, I went for a conference in art. So cardiac, it was European Society for Cardiology, where for lowering the LDL concentration, like say your HDL and LDL, like your low density lipoprofile, they use a drug called statins. So statins lowers your lipid, but it has lots and lots of side effects. So that statin itself has the ability to have many diseases. Ultimately, you're not going to die because of cholesterol. You die because of the statin intake that you are taking it for a longer duration. So, hence we started to look into, is there any other way where we can have a target which targets the particular thing without affecting the other proteins. So that's why we started to work on, we instance our group, a small group where we worked on isoform drug targets. So the role, now the topic of research is the role of channels in cardiac arrest, a cellular level insight for isoform drug target. So we'll see what is the current scenario. The current scenario is we have lots of sudden cardiac arrest that happens in India. So there is three things. One is heart attack, cardiac arrest, and stroke. So heart attack is where you have this chest pain, body aches, shortness of breath, and cold sweat. Whereas in cardiac arrest, it is totally unconscious. You go unconscious, blackout, chest pain, extreme palpitation, stroke that happens in your brain that ultimately leads to a stopping of pumping of your heart. So disturbed in speech, inability to walk, blurred vision. So here we can see that half of the patients or half of the disease is caused by your sudden cardiac arrest that happens in India. So this is the case that we are going to work on. So see here in the Indian scenario, so less than 30 years of age have any coronary artery disease. So most of the people, say approximately 10.5 million people in 2015. Now it has gone drastically. This is a pretty old scenario. Today, this year we'll get the scenario. I'll update you what happens. So going into the cellular structure of heart. So heart. So here you could see the single cell of heart. That's called cardiac myocyte. These are all excitable tissues. So these excitable tissues is the one that gives the information from one cell to another cell, thereby the action potential of cardiac thing gets passed away from one cell to another, thereby the heart pumps. So you could see the lumped up sound. Na? That lumped out sound is because of your pumping of your or the contraction and expansion of your cardiac myocytes. So in that you could see lots of channels. Lots of channels are basically proteins. These proteins play a very important role in passing of your calcium, sodium, magnesium, potassium ion inside and out, ultimately leading to the passage of your current from your first cell to the last cell, thereby the heart works or heart pumps. So this is how the channel protein works. So you have the molecule, this is the sodium molecule, this is being trapped. When it opens, it goes inside, thereby it gives a change in the potential difference. That potential difference is going to play a very important role in opening and closing of the channel. So we'll go to the next slide. So this is the important slide where so you have the sinoatrial node. So you have, I think everyone knows there is four chambers. You have two ventricular chamber and two atrial chamber. So in that first you have the sinoatrial node. So here 0, 1 and 2. Everyone should have seen this ECG profile. PQRST. This is a perfect ECG profile where a typical human being should have. But 99 to 98 to 95% of people won't have this typical ECG profile. So this ECG profile comes from the sinoatrial node, atricular ventricular node, then Purkinje fibers and ventricular thing. So this is basically flowing of sodium, potassium, calcium from outside to inside and inside to outside. That will lead to the action potential. So what we call this as action potential. This Purkinje fibers is what is called the action potential that happens in cardiac. So this happens in your brain also. That is called neuronal action potential. This is your cardiac action potential. So the next slide will explain in detail how the cardiac action potential is translated into ECG. So what we see in ECG is the whole heart action potential, I will say. So the total heart, approximately millions of cells are combined. That is going to give you the cardiac action ECG, whereas it is not millions of cells. So each and every cell has its own action potential. It has the atrial action potential and then it has the ventricular action potential. These two in combined will give you this ECG profile. So the main thing here is the PQ interval, which is less than 0.2 seconds, then ST interval, which is approximately 0.6 to 0.11 seconds, and then QRS complex, that is what the main thing is. So this is approximately 0.7 milliseconds. So this 0.7 milliseconds is going to tell you how the heart is pumping. Okay, so here what does this ECG gives? ECG gives is the amount of sodium, calcium, potassium that goes inside and outside, that will lead to the change in the membrane potential. So each and every cell, so next slide will tell you. So what happens here is the next slide will tell what so this is how it looks. So here you can see it starts from the resting potential is minus 90 millivolt. So once there is that, then it goes from minus 90 millivolts to plus 20 millivolts. So this follows Ohm's law, Nernst equation, everything it follows. So this is a best conductor of electricity. So since there is a flow of ions, this will lead to membrane potential, ultimately leading to voltage current, and the membrane acts as the resistor. So you have from minus 90 millivolt, it goes to plus 20 millivolt. That is your phase 0. Phase 0 is happened by the channel called sodium channels, INA. So sodium channel passes sodium from outside to inside. So you could see from extracellular to intracellular inflow of sodium ion, which is very fast. This happens in a fraction of a second. So in fraction of a second, your sodium channel opens and then lots of sodium ion goes inside. That will lead to plus 20 millivolts. From that plus 20 millivolts, it comes back to the resting state. So that will take approximately 0.6 to 0.8 milliseconds. So 0.8 seconds, sorry, 0.6 to 0.8 seconds, where there is inflow of inflow from inside to outside, there is inflow of potassium ions, outflow from outside to inside calcium ions, and then inside to outside potassium ion, thereby it maintains the resting potential. So this is the basic concept of any excitable cells. Excitable cells present in heart, excitable cells is present in your in a brain, that is your neurons, and then excitable cells is present in your muscle cells, that is your muscle cells, basically muscle cells, heart cells, and your neurons. These three play a very important role in taking the information from the receptor side, and then it passes the information to the brain or heart, thereby it works on. So everything is working based on your sodium, potassium, calcium ions. So inside, from outside to inside it goes, or inside to outside it goes, thereby the potential difference should be maintained. If there is any change in the potential difference, ultimately this will lead to some diseases or a problem in the heart or heart attack, or I won't say heart attack, some problem in the heart. So there are lots of diseases that comes in heart. So these are all the protons that justify or that will pass the sodium. So NaV 1.5, that is the sodium channel proton that passes sodium from outside to inside. CaV 1.2, this is the sodium calcium channel calcium protein or calcium channel protein where it passes calcium ion from outside to inside. So you have the ERG, KR 2.1, KVL, QT, and then MILK. So these are all some of the potassium channel protein where the potassium is passed from inside to outside. Got it? So this movement of ion is the one that is going to give you or change the potential from minus 90 to plus 20 or whatever potential it can. So if there is lots of flow of ions, instead of plus 20, it can go to plus 30. If there is less flow of ions, instead of plus 20, it can go to 0 or plus 10. That will lead to disease. So my research was on particularly sodium channel. So I don't want to discuss about other channels. So sodium channel, it passes sodium from outside to inside. This is the structure of a sodium channel. See, it is pretty big. It's a very big channel. It is approximately 2400 amino acids. So it's a 2400 amino acid and it's a membrane protein. Since it's a membrane protein, it's difficult to get the crystal structure. So sodium channel research has been done for past 40-45 years. Till now, we don't have a crystal structure. Last 2 years, like in 2013, a group from China has found out the structure not through crystal but through electron microscopy. We can't really 100% believe in that structure, but it's a good starting point to work on. So we got the structure of this sodium channel. So this sodium channel, this is a 4-domain protein. So domain 1, 2, 3, 4. So it is having 6 transmembrane helices in each domain, and there is a loop that connects 5 and 6. So this is the pore forming loop. This pore forming loop is very important because if there is a mutation in this pore forming loop, instead of passing sodium, it will pass calcium. Instead of passing sodium, it can pass magnesium. So it is very sensitive. So a few mutation changes is going to pass calcium instead of sodium. That will lead to lots of diseases. Got it? Next thing is your inactivation loop. This inactivation loop is the one that will close. So there is enough sodium that has passed inside. So please close it. So this is the one that closes and thereby potassium from inside goes outside. That is how we get your ventricular action potential. So this pore forming loop, this inactivation gate plays a very important. I don't want to discuss in detail. If I need to discuss, it will go for ages together. So remember, it's a 4-domain protein, 4 subunit protein, which is having 6 alpha helix for each domain. Totally 6 into 4, 624 helices, and it's a 2400 amino acid protein. It's a very complex. Membrane protein. So what is there in the C? So that's A. You have seen B. B is the typical structure, 3-dimensional structure of a membrane sodium channel protein. What is the C? C is the sodium channel protein of a bacteria. C is the correlation between, this is a human one, A is the human one, C is the bacterial one. See the correlation, it looks exactly same, but in humans you have it been expanded into 4 domains, whereas in bacteria or in archaea it is the only one domain which does all the function. So over a period of years, so over millions and millions of years, it has evolved in such a way that it gets complicated. But the main structure looks same. So this is how it works. So you have the open state within 1 to 2 milliseconds. It goes for starting from closed resting state. So from closed resting state, it gets depolarized where it gets open. So then after a few milliseconds, it gets closed, that is closed inactivated state, then repolarized, closed inactivated, and then closed resting. So from resting only it can open. From inactivated state, it can't open. So this is the trickiest part in sodium channel. So that's why you get the lap-tub sound in art. So the sinoatrial node gives the impulse. It should reach the ventricular. So by the time it takes some milliseconds or seconds. So the cells in the sinoatrial node should recover. Thereby the ventricular nodes gets activated, and thereby the sinoatrial node cells gets activated. So from closed state it goes to open state. From open state it goes to inactivated state. From inactivated state it again goes to recovery closed state. Then only it can open. The other way it is not possible. So that is the important aspect of this sodium channel. So this is how it works. So from resting state you have the ions that moves on. So it's an animation. In this Mac it's not working. So in Windows it should work. So I'll pass you the PPT where you can see the animation. So in resting state, the sodium ion goes from outside to inside. There is no movement from outside to inside. In active state, there is movement of ions from outside to inside. And in inactive state, it closes. So this is the basic concept of this voltage gated sodium channel. How this opens? This opens based on the difference in the potential. From minus 90, if it goes to 0 or plus 20, it opens. Otherwise, it can't open. So that's why it's called voltage gated sodium channel. There are two types of sodium channel: voltage gated sodium channel and ligand gated sodium channel. Voltage gated sodium channel opens because of the difference in the voltage. Ligand gated sodium channel opens because of there is a binding of ligand. It can be a small molecule, it can be a protein, it can be a peptide that can bind to the protein, thereby it opens, thereby passage of sodium ions. Okay, coming to this, this thing is another important slide where, so I told you it is sodium channel that does the function. So there are 9 different sodium isoforms. Isoforms are performing the same function, but it is present in different tissues. So I told you it is NaV 1.5 that is present in art. You can say NaV 1.5 that is present in art, whereas there are 9 different sodium isoforms. It is NaV 1.1, 1.2, 1.3, 1.4, 1.5, 1.6, 1.7, 1.8 and 1.9. So 1.2, 1, 2 and 3 is present in neurons. 1.4 is present in skeletal muscle. 1.5 is present in art. 1.6 to 9 again is present in dorsal root ganglion and in central nervous system. What I want to tell here is, see, there is 9 isoforms. If I am going to discover a drug that goes and blocks sodium channel that is present in art. So if that drug is, if there is present, it is present in the bloodstream, it has the ability to go and have an access to the neurons. So if it accesses the neurons, there it is going to block the neurons, or it's going to block the sodium channel neuron that is present there in the neurons. Ultimately, this is going to cause lots of side effects. This is what the concept of isoform drug targeting or personalized medicine. So we are going to go in for the genotyping medicine, where you can find out the genes. So here NaV 1.1, 1.2, 1.3, 1.4, 1.5, 6, 7, 8, 9, 10. The 8, 9, we have found out there is another isoform called 10. So you could see 1 and 2 is responsible for epilepsies. 4 is responsible for paralysis. 5 is responsible for many diseases caused in art. 6, not much work is done. 7 and 8 is responsible for pain. So in 2009, when I was doing my PhD, there was an article in Nature. So that created lots of stir. So they found out there is a patient, or there is a three humans in Pakistan. So they can go into the fire, get burned, but they don't feel the pain. That was very much interesting. When they analyzed, they found out that the gene or that the protein that is responsible for it is NaV 1.7. So there was a 3 nonsense mutation. That mutation has created insensitivity to the pain. So you don't feel the pain. You go get burned, you don't feel the pain. That's pretty interesting. So if we can target, or if we have any molecules that can target NaV 1.7, you can be a billionaire, or you can be a you can be a very good scientist or Nobel laureate where without even disturbing other sodium channel, if you can target only NaV7, I can destroy the pain. And another thing is, say most of the audience are women. If you go for cesarean or C-section, the drug or the painkiller that gives that you get it in the spinal cord is the one cause of many side effects rather than the C-section itself. C-section gets cured, but the drug that's been injected for pain is the one that has lots of side effects. So the research companies or the pharmaceutical companies went for a hunt to find out is there any small molecule or peptide or protein that has the ability to only block NaV 1.7. Still now nothing is being discovered, but many are in pipeline. So many are in pipeline. Till now nothing's been released. But if something is released, so they are going to be the billionaire. Okay. So likewise, you can target only NaV 1.5. If I can target only NaV 1.5, I can block only the art. If I go to target only 1.4, I can block only the muscle or paralysis. So if I can block only NaV 1.1 or 1.2, it's going to have a effect on epilepsy. So this is how the research went on. So this is what is called isoform drug targeting. So since I am working only on sodium channel, I am going to give only on sodium channel. Likewise, we can do it for calcium channel. Likewise, we can do it for potassium channel. Likewise, you can do it for magnesium channel. Likewise, we can do it for chloride channels. So there is lots of scopes we can work on it for each and every. If we can find out a particular isoform targeting drug, you can be a good researcher followed by a millionaire. So this is what. So next thing is. So this is how. This is one of my research in my PhD where these are all some of the mutations that causes diseases. So so the green color one is atrial fibrillation. So palpitation. There is a. These are all in cardiac sodium channel. Brugada. Then you have the sudden infant death syndrome. Then you have the long QT syndrome. You have seen the QT spike now. So if there is a long, it's called long QT syndrome. So these are all some of the mutations where these mutation is going to cause the disease. So if there is a mutation in any part, see, it's not only prevalent in only one part, it is prevalent everywhere in the protein. So this is telling that not only a particular part of a protein is responsible, it is all the part of the protein is responsible for the functioning of the thing. So taking this, I worked on, the group worked on, we targeted toxins. To start with, we targeted 3 toxins. One, the toxins from scorpions. The second, the toxins from cone snails. Third, the toxins from snakes. So this is there. This is my. So those who know me knows my research. So they call me as the snake man. So we have snakes, we have toxins, we have venoms. So my lab there in Banariaman had snakes all time. So 8-9 years we had snakes in my lab itself. We extract venom, we do research. That's all happens. So what is the speciality of venom? So venom is a complex molecule. So venom is a complex molecule. It has lots of small molecules. It has lots of proteins. It has lots of peptides. So these peptides can be the target for particular sodium channel. So likewise, we started to work with Rim and Balkis from Institute of Pasteur, Tunis. So this was an Indo-Tunisian bilateral project where we took thing from scorpions, the venom from scorpions, because that LVMT lab in Tunis is the lab which produces anti-venom for all the scorpion toxins all over the world. That's the only lab, approximately 80 to 90% of the anti-venom that is being produced or that is being used are from this University of Pasteur lab. So we started with our Indian scorpion. We have 3 main scorpions. This is the Swarmadomi. This is the largest scorpion species. So this has the venom. So anyone guess what is the price of this venom? It's a very precious. It is much more precious than your gold or platinum. So 1 milligram of this venom cost 48,000 rupees. So 1 milligram of scorpion venom costs 48,000 rupees. So you can't get like that. So you need to get the approval from the government, go get it. There's only one place in Chennai, you get it from the Crocodile Bank. So we need to get approved and get the venom. So it is pretty easy. If you get these scorpions, it is easy to extract the venom. So these are all the scorpions that we worked on. So what was the researches? So the researches, so see here the sites where the selectivity filter is the site for toxins. So the selectivity filter varies from NAV 1.1 to NAV 1.9. So not very much variation, but there is a slight variation. So each and every toxins goes and binds to this site. So if it's possible to find out a peptide or a small molecule which can particularly block any particular isoform, that will be a breakthrough research in toxin. So we started with our scorpion toxins. So we found out some small molecules and some peptides. So I don't want to reveal these, reveal the sequence or the molecule, but these are very much interesting. That will have that has an effect on NAV 1.5. We didn't find it for pain, but this is being used for cardiac sodium channel. So this is one venom. So this is the next we worked with Indo-Austrian project where we took the venom from cone snails. These are called conotoxins. So I don't think most of the people who know me knows what is conotoxin is. For the people who don't know me or like who haven't followed my research, I will put a video about conotoxin. It's a just 4-minute video. Find out how lethal is the video, how lethal the toxin is.
Crawling along the ocean floor is a cone snail. It looks harmless, but appearances can be deceiving. 4 tubes jut out from its shell. The long one at the top is a siphon, which it uses to inhale water and detect prey. The smaller ones on either side are its eye stalks, and in the middle, a deadly proboscis. Concealed within the tip is a lethal weapon. A harpoon loaded with a neurotoxic mix so complex there is no antivenom to thwart it. It's armed with at least 100 different toxins, more than any other creature. When some cone snails go hunting, they like to burrow into the sand and wait. When a meal happens by, the cone snail fires its harpoon. The poisons quickly paralyze the fish. Once its prey is helpless, the cone snail retracts it through its proboscis until it engulfs the fish, swallowing it whole. The snail is no longer hungry, and its appearance no longer harmless.
Yeah. To start with, this is the cone snails. This is the cone snails we call in Tamil as kumbu nattaigal. So these are all not on the shores. These are all on the deeper oceans. They are in the bed. So along the coastal Tamil Nadu, itself, we have approximately 270 different species of cone snail. So there are 270 different species of cone snail. They are very colorful. They are very, very colorful. So they come in the when the fisherman goes for fishing, they come as thrashes. They come as thrashes. Approximately 270 to 280 varieties of cone snail is being discovered in found along the coastal Tamil Nadu itself. Yeah, the lethality of this venom is in such a way that it can or it has the ability to kill a dinosaur. So it has the ability to kill a dinosaur. So I'm not exaggerating. It is that kind of venom it gives. It gives a cocktail of peptides, cocktail of small peptides, which is going to target the nervous system, ultimately causing death in a fraction of a second. The death happens in a fraction of a second. So what this is going to do in the research that we are going to work for painkillers or the drug for art. So as I said, it's a cocktail of venom. So if we can isolate a particular peptide from that cocktail which targets that particular channel, then you will be a successful researcher, because that is going to cause pain or kill pain, ultimately leads to death. It's only one peptide, not the cocktail. If the cocktail is given, it's going to cause death. If you are going to segregate that peptide or cocktail, get one peptide or one small molecule, that one peptide or one small molecule, ultimately that will be a isoform targeting peptide. So this video you have seen. So this is how the cone snail is. Its cone snail is of. So what I'll show some of my collection. So these are all some of the cone snails that I have collected. Started with Conus tessellatus. Yeah, is it audible? Conus tessellatus. Then we have Conus amadus, Conus molli, Conus emberimus. So see the pattern, the color pattern. The color pattern keeps on changing. These are all cone snails. The color pattern gives you the cocktail. So each and every species gives each and every species gives each and a cocktail of venom which is lethal to the core. So what is our research? See, this is the Conus lawrenci, Conus bayani, Conus figinalis. Could you see the bigger version that's there in the gloves? So that is the Conus venom duct, that big. This approximately 20 to 30 centimeter, followed by a bulb. You have a big bulb. It's a respiratory for all the venom, and that venom is being pumped in and then is being given to the prey. Each and every prey receives a different cocktail of venom. That's the speciality of this Conus species. So what we did is we analyzed all this. We started to analyze the Conus amadus, Conus monile, Conus embericus. Each and every species has a different cocktail of venom. So the problem right now we have in this research is, so there is, we did the Maldito, we got some results. In that results, there are similar peptides that can't be isolated. So similar peptides in sense, they have the same molecular weight, mass by charge ratio. Say if I have a peptide of 5 amino acid sequence. So 1, 2, 3, 4, 5. So that's the 5 amino acid sequence. But this, or it is 4, 5, 3, 2, 1. So what combination of this peptide is present in this species? We couldn't get it. So that is where our research is in the thing. So most probably we'll be having a special HPLC. If we can isolate the particular small peptide, then it will be a good breakthrough. So we did the analysis with conus monyl and conus amadus on NAV 1.5 and NAV 1.7. Conus amadus has an effect on NAV 1.5, whereas conus monyl doesn't have an effect on NAV 1.5. Conus monyl have an effect on 1.2 and 1.7, whereas conus amadus doesn't have it. So these are all the small peptide analysis that we did, but we doesn't know what is the sequence of this peptide. So these are all cysteine-rich peptides. So we need some more funding to get through and then do peptide analysis of these molecules. So this is one research. So next we did some molecular dynamic analysis on the various voltage gated channels to find out how this binds. So we found out with NAV 1.4, this is mu G3A, this is the conotoxin from conus amadus. So they found out these are all binding to the loops connecting D1 and D3. So D1 and D3, if you can go to the first slide, that will tell you the D1 and D3. What is D1 and D3? This is D1 and D3. So here pore forming loops. So this plays a very important role in this. So we found out there are some residues. Some residues, namely, many are hydrophobic, many are hydrophilic. Some are, to my surprise, there are some proline residues which play an important role. Only one proline residue plays a very important role in blocking. So these are all some of the findings that we found out. So see, this is lysine, arginine, aspartic acid, glutamic acid. So most of the positively hydrophilic amino acid plays a very important role in binding. So I don't want to go in detail. I think I'm overdue. Like I should have stopped by 6. So we do research on other toxins. Say I do research on bee venom. Bee venom is a very important venom. That I didn't put it like bee venom. So I don't know whether you have read an article during COVID times. In China, those who have apiary, like apiary. Apiary is the, where they breed bees for honey. So usually these people will be bitten by this honey bees. So that people are not contracted with COVID. They did research that this venom has an antiviral property. They haven't characterized. There is a main protein called melatonin. So that will that has an anti-COVID properties. Then we did analysis on this COVID we did the melatonin on the corona spike proteins. So it has a very good binding. So that is one. Then we did little bit of research on this. We have done research on cobra and on crate. Crate is a very important neurotoxic venom. So this is also very expensive venoms. So approximately, this is the lab where we work on. So this is the lab in Banariaman. This is the lab in University of Vienna. Some of the equipments that we work on. So the main problem or the main thing is none of them are worked on this venom. First thing is it is expensive. So 1 milligram of cobra venom costs 40,000 rupees. 1 ml or 1 milligram of crate venom costs approximately 56,000 rupees. 1 milligram of crate venom. So if there is enough funding and these things, we can go to the next level of research in venom peptides. So that's why we started with bees. Bees are pretty cheaper. We cultivate our own bees. We get the venom. That is also pretty hard to extract the venom. The venom from bees is very, very, very small, minute. So there's lots of things to talk. Since there is no time for me to talk, I would stop here. I would answer you any questions if possible. Thank you.